I'm here to talk about, we get to pick our own topics, so I'm here to talk about clinical research in Parkinson's disease. And I've been here since July 1st of 2016, but before that I spent 30 years in practice at Rush University Medical Center in Chicago, where we had lots and lots of clinical trials for Parkinson's disease. And clinical trials for Parkinson's disease are very rewarding for the investigator, for the researcher, because these are the trials that lead to new treatments. So they make the lives of people better every day, and that's what I'm in this business for, making the lives of people better. And I'm fortunate to have participated in the research that supported every single drug that's on the market now for Parkinson's disease, every single one except levodopa, which was in the late 60s. So I was in grade school, so I couldn't really participate in that research yet. So this is really a talk about what clinical research is all about. And before I came here, UW really didn't have any active clinical research trials for Parkinson's patients. And there's a big, long story that goes with that has to do with politics and institutional initiatives, et cetera. But one of my goals was to change that. And so we've had one trial already approved that's no longer recruiting subjects, and then 2 more that are about to be approved. And I'll talk about those at the very end. So really, I'm going to talk about how clinical research fits into the overall scheme of research in Parkinson's disease. And this actually applies to other diseases as well. So if you're a care partner and you have breast cancer or congestive heart failure or arthritis, the same kind of principles of clinical research will apply. And then we're going to talk about some of the kind of pitfalls of the media representation of clinical research in Parkinson's disease. And as Nancy said, I'm willing to take questions at any time during the talk. So if it seems like I've gone off track or you're not understanding something, please do feel free to shout out a question. So the title of this is "A Bird Cannot Fly with One Wing," and that's really because participation from patients is required for us to make any progress in Parkinson's disease. Is this not going forward? It's what?
Yeah,
it should be that. There. Oh, you need the magic touch, the healing touch. So one word, I always have to start with a little bit of a pitch. Seriously, I can't do this. Oh, okay. And then I can't go backwards. Okay. Animal research saves lives. I always have to just put a pitch in for animal research because animal research is under fire in the world. It's under fire in this country. And it's important to understand that no new medicines will ever be invented without animal research. So just remember, if you hear a lot of talk about how animal research is not necessary and you can use computer modeling, none of that is true. We are still dependent on animal research to make the progress that we need to make. So we're going to talk, as I said, about a little bit about the Parkinson's basics, just to give you the framework for the research, a little bit about treatment, and then preclinical research, and then clinical research. So first of all, Parkinson's disease is really a complex disorder. So we think about it in terms of the core features, which are tremor, rigidity, or increased resistance when we move your arms and legs around, bradykinesia or slowness. Those are kind of the core features of Parkinson's disease. But everyone in this room knows they're not the only features of Parkinson's disease. So there's a whole bunch of symptoms that we call non-motor symptoms. They aren't related to abnormal movement, and they're things like constipation and bloating and gas production and bladder problems and fatigue and pain or numbness and treatment side effects like impulse control disorder, gambling or shopping, for example. Problems with sleep, which are almost universal in Parkinson's disease. It's neuropsychiatric, things like hallucinations, depression, anxiety, cognitive, things like either mild memory loss all the way to dementia. And then these things all exist in the soup that is the rest of our bodies, and so all the other comorbidities that happen with aging, things like diabetes, ulcers, inflammatory bowel disease, bacterial overgrowth of the intestines, cardiovascular disease like heart attacks or congestive heart failure, osteoporosis, which is increased in frequency in people with Huntington's disease, and high blood pressure. So it's a very complex disease nested in an aging population, which has all its own other comorbidities. So let's just talk about the disease itself, because the motor symptoms that you heard about, the tremor and the rigidity and the slowness, are things that happen as a result of the disease, but there's quite a bit of water under the bridge before that happens. So this is what a brain cell looks like. Brain cells are called neurons. This is the cell body where the nucleus is. That's where all the genetic material is. And this is the axon. So you might think of cells as being just little round guys, but nervous system cells or neurons have these very long processes. So for example, the sensation in the tips of your toes, that nerve process goes all the way from the tips of your toes all the way to your spinal cord. It's a very long, one cell goes all the way that way. And so what happens a lot in brain function is you have a lot of the action going on here, and so brain functions happen by chemical release and chemical actions. So the chemicals will be manufactured here in the cell body, and then they'll travel to the end of that axon and then be released somewhere and then cause an effect. I got to point this way probably, right? So what's happening, the very first thing that happens in the nervous system is that these neurons, these brain cells, start to accumulate a protein. That protein is called alpha-synuclein, and it's not protein that you eat and then goes to the brain cells. It's protein that's made within the brain cells. So the brain cells make all the proteins they need to repair themselves, to do the chemical reactions that happen inside, to make the chemicals that are used for communication in the brain cells. So what happens is that there's a mismatch in the manufacture of that protein and the recycling of that protein. So too much of the protein starts hanging around the brain cells. You know, when you hang around with the wrong kind, bad things happen. So the protein starts to form clumps. And then those clumps start to interfere with lots of the things that normally happen within the brain cells. Honest to Pete.
Are they just clumps of protein?
So good question. So are they just the protein clumps of alpha-synuclein or mixed with other things? So they tend to attract other proteins. So like the Alzheimer's protein gets kind of glommed in there, and other proteins do. And it interferes with the function of those proteins now. So now you've got a protein that's not working, and it's congealing up in the cells, and then it's also interfering with other proteins, collecting other proteins, et cetera. And it starts to interfere with all the processes that happen in the cells. So these are some of the things that go wrong. So oxidative stress, think of that as brain rust. Rust is oxidation of metal, right? So you get kind of this damage to the cells by hungry oxygen molecules. The cells all produce their own energy in a structure called the mitochondrion, and that doesn't work very well. So now the cell's not producing enough energy. And you can imagine a cell that's not producing enough energy can't run its own chemical reactions, also can't power the protein manufacturing apparatus. A lot of problems, and the most important thing is cells in the nervous system have a charge on their outer perimeter. It's like an electric fence, and it helps to protect them. And when the energy starts to fail, you don't have that protective electric fence, and so now the cells become vulnerable to attack from other things in their environment. And then there's something called excitotoxicity. Some chemicals are excitatory in the brain, and some are inhibitory. And too much excitation in the brain actually causes calcium to rush into the cells. And calcium, which we all think is a good thing, is not a good thing if there's too much, and that causes cell damage. And then there's failure of transport, so the cell can't move products back and forth. Interference with other genes, so some of the other genes that make the cells do what they're supposed to do don't work. And then finally, the protein abnormality can interfere with other proteins. So what happens is kind of a multiple failure of normal cellular processes. And then that causes the cell to die. And the reason that happens is brain cells are very sophisticated, and they try to repair themselves when they're under stress like this. But a brain cell, if it can't repair itself, it activates a suicide pathway, so it kills itself. So what happens is what's called apoptosis, or cell suicide. And so these dopamine cells, which are right here in this area where the little red blobby part is, that's where the dopamine cells live. And you've probably heard in the clinic that the movement problems of Parkinson's disease result from too low dopamine. Has everyone heard that, right? The dopamine is too low. So what happens is the dopamine-producing cells, they're shaped like this. The cell bodies are here, and here the long axons go up to other parts of the brain. As the cells die here, they can't send that dopamine to the other areas of the brain, and that's what causes the problems, mainly the movement problem of Parkinson's disease. Does that make sense? Okay. So then what happens is, so you've got this protein collection, dopamine neurons die, too little dopamine, and there you get those characteristic motor features: tremor, rigidity, slowness, and what's called postural instability, which is a sign of more advanced disease where you have problems with falling. But that's not the whole story. And the reason it's not the whole story is that there are other brain cells that are involved as well. We focus a lot on dopamine in the clinic because we can help dopamine. But other brain cells are also involved. And those brain cells don't necessarily work by dopamine. So other neurochemicals, other chemical messengers between the brain cells start to become decreased. And they cause non-motor Parkinson's disease, which is all the other problems: fatigue and sleep disorders and problems with thinking and memory and bladder frequency and constipation and pain. All those things, anxiety, depression, all those things are caused, in fact, by other brain cells dying, and they don't involve dopamine. And so the treatments that we normally use to make people walk better or move better or stop shaking don't typically help any of those other symptoms. And that's important because we aren't very good at managing those symptoms, and they cause a lot of disability too. So one thing that's been clear just in the last 15 years or so is that, and this has been research that's been done using autopsy of people with Parkinson's disease. And this is when, so remember the dopamine cells are right here. And what happens is when those cells start to go, then you start to have these movement problems. But before that happens, over here, you start to lose brain cells lower down in this stalk of the brain, lower down. And this is called prodromal or premotor Parkinson's. So when you get your first sign of Parkinson's, come to the doctor. It's either a tremor, or your handwriting's too small, or you've noticed that you don't swing your arm, or maybe your walk has got a shuffling quality to it. By the time you get to the doctor, you've got degeneration here, but also you've got degeneration lower down in this part of the nervous system. And so there's like a constellation of what are called prodromal or premorbid symptoms that include things like being excessively sleepy during the day, being constipated, not having a normal sense of smell. There are all these kind of changes that are happening in those years, up to 10 years before you're diagnosable, that are caused by actually, there are brain cell death lower down in the brain, and then it gets to, just kind of ascends, gets to this point, and you start to have tremor, rigidity, slowness. It doesn't stop progressing then. It continues to progress up the stalk of the brain and then all the way into the upper parts of the brain, and that's when people tend to get problems with falls that don't respond to medication, problems with thinking and memory. Those kinds of changes happen because of the progression. So ultimately, if we're going to find something that cures Parkinson's disease, two things are going to have to happen. We're going to need to identify people earlier in the disease course, before they come in with their tremor, their rigidity, because you've already got degeneration here. And then we're going to need to be able to find something that prevents that kind of spread of disease across the brain. Does that make sense to everybody? Okay. So I did tell you that dopamine is the chemical that's responsible for this tremor and the slowness, the kind of core features. Interestingly, it's also responsible for changes in color vision. So people with Parkinson's disease don't perceive colors as well as people who don't. And also visual contrast. So if you don't have a really high black to white, like if you're reading the New York Times, which has a gray background with black type rather than a white background, it can be hard to read because the contrast perception isn't very good. And that low dopamine also contributes to a drop in blood pressure when standing up. So those are kind of the core low dopamine symptoms. But other chemicals are involved as well. So norepinephrine, we think of that as adrenaline, the fight or flight chemical. That's also affected, and it can be affected before the dopamine is affected. And that causes depression, a very characteristic sleep disorder that precedes Parkinson's disease in many cases, which is called REM behavior disorder. Has anyone ever heard of REM behavior disorder? REM behavior disorder is a sleep disorder where you act out your dreams, because unlike normal, you're not paralyzed when you're dreaming. So the normal system, you're paralyzed during dreaming, so you don't run out of the house. But in people with Parkinson's disease, often years before they develop the first movement problems, can have this dream enactment where they punch, they kick, they fall out of bed, break the lamp, And then if you wake them during that spell, they'll remember that their dream is that kind of violent dream. So it's a very characteristic condition. Not everyone has it, but it's very suggestive of the future development of Parkinson's disease. And then this norepinephrine changes also contributes to the slow standing blood pressure. Acetylcholine also involved in REM behavior disorder. It's the main chemical that's involved in cognitive impairment. It's the same chemical that's involved in Alzheimer's disease. Dementia is involved in Parkinson's disease, cognitive impairment and dementia. Acetylcholine is also involved in sense of smell, so that's one of the reasons for the loss of sense of smell. Also contributes to the standing blood pressure drop, is definitely related to poor swallowing, and all of the gastrointestinal stuff. So the gastrointestinal tract is slow from the mouth to the anus. Everything is slow to swallow, slow to move food through your stomach and your intestines. So the acetylcholine deficit causes poor swallowing, bloating, gas, and constipation. And then serotonin is one of the happy chemicals. So if your serotonin's affected, you have depression. It also contributes to this REM behavior disorder. And then glutamate is also involved in the cognitive impairment, decreased balance, and slowness. And unlike dopamine, where we have really good medicines to replicate or mimic its effects and really good medicines to increase the amount of dopamine in the brain, there are not very good treatments for these other chemicals. And that's why we're not quite as good at managing the other aspects of Parkinson's disease apart from those core motor features. Oh, please, there we go. So I'm going to talk a little bit about the basics of treatment just as a way to understand the research of the new treatments. All right, so for the dopamine responsive symptoms, there are really only 3 ways to fix them. So the way that's been done since the late 1960s is to increase the amount of dopamine in the brain. You do that by giving levodopa. There are a bunch of different forms of levodopa. It's all levodopa. Or you can actually, the brain works by chemistry and electricity, and sometimes you can replicate a chemical effect by using electricity. So there's an increased use of stimulation of various brain structures for various diseases, not just Parkinson's. So deep brain stimulation is kind of an electrical way to replicate the same changes that occur in the nervous system as change with increasing the dopamine. And it's important to understand that because it doesn't give you anything that the medicine doesn't give you either. So it only really reproduces what you get from the medicine. And then the other thing you can do is you can use medicines that actually stimulate directly the dopamine-sensitive structures in the brain. So where that dopamine is released from the long terminal of the nerve cell, it acts on what's called a receptor, which is specialized to respond to the chemical. And there are medicines that directly stimulate those structures in the brain. They're called dopamine agonists. Opposite of antagonist is agonist. So carbidopa-levodopa is the absolute best medicine for Parkinson's disease. Don't let anyone ever tell you that it's not. It is the best medicine for Parkinson's disease. It's the cheapest, it's the most effective, and it's the most cost-effective and has the fewest side effects of any medicine. And the reason is because it gets turned into real-life dopamine. It's the real deal. So you swallow it, and then it goes into your stomach, but it's not absorbed in your stomach. It has to leave your stomach and go into the small intestine, which is here. It gets absorbed across the small intestine. It goes into the bloodstream. And then, so it's in the bloodstream, and then it has to get into your brain. And there are some challenges there. So it gets into the bloodstream by going through with a carrier. So it has to be kind of carried across the wall of the intestines. It's the same carrier that carries the protein building blocks across the wall of the intestine. So if you've eaten protein at the same time as your medicine's in there, there's going to be competition for those carriers. So there are a lot of things that we can do to kind of make sure that you get better absorption. Once it's in the bloodstream, it's prone to being turned into dopamine in the bloodstream. So levodopa is not dopamine. Levodopa is what's called a precursor. It's one chemical reaction away from dopamine. And the reason we have to give levodopa instead of dopamine is dopamine doesn't cross into the brain. The brain has a very sophisticated way for keeping things out. It lets certain things in, like alcohol goes in like nobody's business, and cocaine goes in there like nobody's business. But a lot of drugs, especially big drugs, can't get in there. And and dopamine is one of those. Dopamine doesn't. And dopamine in your bloodstream causes side effects. It causes the drop in blood pressure. It causes nausea and vomiting. And if it can't get into your brain, it does you no good. It's interesting that, so carbidopa, levodopa is how dopamine is sold in this country. And that's because carbidopa allows more of it to get into the brain by preventing it from being turned into dopamine in the bloodstream. But still, 90% of that swallowed pill does not get into the brain. Only 10% gets into the brain. But that's enough. And it has to go through this blood-brain barrier. And then it goes into those brain cells that I told you about that make dopamine, and then those brain cells will turn it into dopamine. And then a miracle occurs because it improves all the symptoms of Parkinson's. Does that make sense? Anyone have any questions about this? And so virtually all of the medicines that are on the market the goal is to get more dopamine in the central nervous system. It's very simple. So you got a bunch of medicines. They're all on this slide. Okay. So here again we have the blood-brain barrier. If the levodopa turns into dopamine in the bloodstream, I should move this to the other side, then it won't cross the blood-brain barrier. So the carbidopa that I told you about prevents the levodopa from being turned into dopamine. There's also another enzyme that breaks down levodopa called COMT, and the medicine entacapone, which is Comtan, and it's also in Stalevo. That medicine blocks that enzyme, and it's also a drug called tolcapone that's not used very often, blocks that enzyme. So if you take carbidopa and entacapone or tolcapone, then more levodopa gets into the central nervous system, but still the vast majority doesn't get in. Once it gets in, it's supposed to get turned into dopamine and then go to those dopamine receptors and cause the symptoms to get better. But there are two other enzymes here. So there's monoamine oxidase, which breaks the dopamine down into something that's not effective called DOPAC. And then here's catechol-O-methyltransferase, COMT, again, the same little culprit from down here, causing it to be broken down again into an inactive component. So the drugs selegiline and rasagiline, people have heard of those, commonly used adjunctive medicines. They block this one enzyme, so they increase the dopamine in the brain. And tolcapone here blocks this catechol-methyltransferase, again, so that more dopamine is in the brain. The dopamine goes and acts on its little receptors, and all happiness breaks out. And then the other medicines, the other main symptomatic treatments are the dopamine agonists that I told you about. So these are built to look like dopamine, basically. They're chemically made. And there are three. There's pramipexole and ropinirole, and then there's rotigotine. And rotigotine is the one that's available in a patch form. And what these drugs do is they just directly stimulate the same receptor. So they mimic the effects of dopamine, but they aren't real dopamine. And the brain is a very smart place, and it knows it's not real dopamine. So they're not quite as effective. So they do help all the symptoms, but not to the extent that levodopa does. So that's the whole of Parkinson's disease therapy in a nutshell. And there are a bunch of different preparations of levodopa. There's controlled release levodopa. There's extended release levodopa. There's Stalevo, which is carbidopa and levodopa and entacapone. There's just a bunch of different ones. And there's some in the investigational stages, one that you inhale and one that you can put in like a insulin pump. It goes into it's a little pump, and it goes to a needle that goes under the skin. We'll talk about that briefly at the end. But all of those still have to, once they get in the bloodstream, they still have to go through the same process of becoming, stimulating that receptor. Any questions about that? Yes? Oh, so that's great question. How does stress make your symptoms worse? The reason is because stress instantaneously changes the balance of your brain chemicals. Instantaneously, so it ramps up the adrenaline, which causes the tremor to get worse. So it's the kind of instantaneous changes in chemicals. And certain of the symptoms, the one that is most sensitive to stress is tremor. Tremor is what we call state dependent. So depending on what your mental state is, the tremor can just go like gangbusters. But all the symptoms are somewhat stress related. But it's important to understand that the stress doesn't make the disease worse. The stress affects the symptoms of the disease. So, but it's a great question. Oh boy. So that's kind of the symptoms. So all of the new treatments that are coming out are some modification of levodopa or some way of trying to engineer those other reactions that are happening to either get more into the brain or let it last longer when they're there. And very little, actually, of the current research is involved in looking at new treatments for the non-motor aspects of Parkinson's. So the people are interested in finding better treatments. But it's not really something that a lot of drug companies are investing a lot of money into. And they do cause disability, so it's an important target. Now, so that's kind of symptoms. But remember I told you that there's the disease, and then there are the symptoms of the disease. And if we're to find a cure for Parkinson's disease, it's not going to be better symptom control. It's going to be understanding what's happening, the biology of the disease, and finding a way to intervene in that biology to make things better, to slow it down or reverse it. So this is just a slide showing kind of what we do with preclinical research. So here's one way that animal research is really critical, is we model the disease in animals. And then as you model the disease, then you learn what's happening in the cells to make them sick, basically. And so here are 4 different targets. I'm sorry, when these were wide format. When I made them smaller, everything turned microscopic. So one target is the alpha-synuclein. So that is the premier thing, right? It's the first thing that's happening is that protein accumulating in the brain cells. So one way to avoid that would be to vaccinate you, for example. Use your immune system to try to clear that protein out. And so that's an area that people are investigating now, of giving a vaccination and then having your immune system go and kind of scavenge out that protein. There have been similar studies in Alzheimer's disease, and unfortunately, if your vaccine is too strong, then you get a lot of inflammation in the brain, and inflammation in the brain is not very pleasant. So this is kind of being refined and reworked, but it's an interesting strategy. Another thing you can do here, so there's a whole system for getting rid of proteins in the cells. It's called the proteasomal system. And you can find medicines that kind of ramp that up. And so these are natural scavenging kind of mechanisms in the cells. So that's another kind of target. And then neuroinflammation. So this is a topic that's become kind of popular lately. It's kind of come and gone in the research community about whether there's actually inflammation. So inflammation would mean the infiltration of inflammatory cells, things like antibodies that are causing a lot of damage to the tissue. So that's a very interesting target right now. And then finally, this mitochondrion. So the mitochondrion is that energy factory within the cells, and we know that it's failing. And so strategies to improve mitochondrial function. So the purpose of preclinical research, especially animal research, is try to find out new targets. So you identify a target, and then you try to find a treatment that interacts with that target in a way to reduce the impact on the disease. So that's really when we're talking about trying to find real primary treatments of the disease, that's what you have to do. You have to do the work, identify the targets, and then try to find things that will work on those. Any questions about this? Yes? So great question. No animal gets Parkinson's except a human. Remarkable. So when you model the disease, there are various ways. So you can poison the brain, either by injecting a poison into the carotid artery on one side. That's called MPTP is the poison. That's been a kind of mainstay. Or there have been some recently described very rare genetic variants of Parkinson's disease where you put the gene into the animal and model it. And then probably the one that's going to turn out to be really important is you can actually ramp up the animal's production of alpha-synuclein. So then you get the same kind of a recapitulation of what's happening in the people where the synuclein starts to accumulate. But if you're thinking, what I think you might be thinking is, how do we know that those things are relevant, right? Since no animal gets Parkinson's disease, and since no one put poison up any of your carotid arteries, how relevant are those models? And it's a significant concern. But unless animals, and they're not going to get Parkinson's disease, it's a uniquely human disease, which is interesting in its own right. Any other questions about that? So that's kind of everything that happens before the clinical research, but I really want to focus on clinical research today. So, and specifically clinical trials. So a clinical trial is any research study that prospectively, so in advance, assigns human participants, that's people, or groups of humans to one or more health-related interventions. And an intervention can be a drug, it can be a surgery, it can be an exercise, it can be some other, anything could be the intervention. And then the purpose is to evaluate the effect on the health outcomes. So that's the World Health Organization definition of those. And it can be drugs or cells, surgical procedures, devices, behavioral treatments, changes in how you care for people, or prevention, primary prevention. So that's kind of the definition of clinical research. Now we're back to our cartoon at the beginning. And what I've done here is I put on the top symptomatic and neuroprotection. So symptomatic, that's what I just told you about. That's ways to increase dopamine activity in the brain, basically. And so that's what all this stuff is. Anything that has to do with dopamine or surgery, those are all symptomatic treatments. They improve the symptoms, but they have no effect on the disease. And then anything back here, so if you can deal with, get the synuclein out of the cells or deal with any of these downstream things that happen, then, and prevent the death of dopamine neurons or other neurons, then that's a neuroprotective intervention. So we characterize research into neuroprotection and symptomatic treatments. Any questions about that? Excellent. But we can't do research without participants. And those participants are you and people like you. So clinical research studies are how new knowledge is generated, and they help to find new treatments. And being a research subject is not for everyone, but unless people do volunteer for research participation, we will not find a cure for Parkinson's disease. 100% guarantee, we will not. Now, a lot of times people ask me in the clinic, how many years? When will there be a cure? How many years? And I understand why that's the question everyone wants to know the answer to. But it's impossible to answer, and the reason is because if we found the cure today in a laboratory, if it's a new compound that's never been studied before, it's going to take 16 years to get it to the market. That's a long time. There's a new initiative to try to repurpose old drugs, and I'll talk a little bit about one of those later, where you don't have to have all the discovery, and a lot of the safety testing has already been done. So that's actually faster. That you can usually do in about 6 or 7 years. And then the other thing that's very sobering is if you start with compounds that are screened. So drug companies have very big operations to screen. They have a target, something they want to do, and then they screen a library of drugs, and they find one that works for their target. For every 5,000 to 10,000 screened compounds, 250 are considered good enough to go into animal testing. Only 5 of those will survive to clinical trials, and only 1 will be approved by the Food and Drug Administration. So it's a huge funnel that's constantly going to try to find these new treatments. But it's very sobering, I think, how much work has to go into a new treatment. But it tells you how many people are interested and how much money is being invested in finding a cure, too. Now, I can't just set up shop and do a research project, thank God. And no one can do this. There has to be some kind of oversight. And so most of the oversight, most of the kind of new drug, all the new drug development is overseen by the FDA, Food and Drug Administration. And then there are various other federal organizations that have to do with protection. So this here, Office of, this is protection of research participants, for example. And then at the local level, there's something called the IRB, which is the Institutional Review Board, and that reviews any clinical research protocols. If I have a great idea, I have to write it up and submit it to them, and they'll come back and say, well, this isn't a very good idea at all because X, X, and X. Or, and they're the ones who oversee that the research at University of Wisconsin is done in an ethical fashion. And then there's the sponsor. So often in this country, it's a drug company, or it can be the National Institutes of Health, which is your taxpayer-funded medical research organization. Other funders, so the Michael J. Fox Foundation might fund a trial. And then the principal investigator is the person who writes the whole project. And then there's often a data safety monitoring board that's people who are not involved in the research except to make sure that all of this potential side effects are being screened for. And they're very important to protect the safety too. So there's an awful lot of hoops to jump through. And don't get the idea it's any different in any other country. Every country has a kind of an oversight. Canada is one of the most difficult, in fact, in all of the EU countries. So it's not just the United States that has such a cumbersome oversight process. You may hear about the phases of clinical trials. In order for a drug to get approved, it has to be proven to be effective in phase 3 clinical trials. A phase 1 clinical trial is the very first study in people. It's sometimes in healthy subjects or in a few people with the disease, and it's looking at things like what doses can be given safely, what kind of side effects can we expect. And then the drugs that survive the phase 1 go into phase 2. A phase 2 study is usually looking at the effects of different dosages and getting a little bit of a signal about whether we can expect any benefits. And a phase 3 study is a large study. Usually in a Parkinson study will involve hundreds of subjects, especially for a neuroprotective clinical trial. And in those studies, the subjects are randomly assigned to either the intervention, whatever it is, or to an identical appearing intervention that's a placebo. So it doesn't have any active intervention in it, but it looks, tastes, smells, feels, everything just like the other one. And the reason for doing that is the brain's a very powerful place, and if you give a person a pill, even if it has nothing in it, that person will feel better. It's a great thing, but when you're looking at research, you have to have that comparison group to know if the changes in your treatment intervention group are real, or whether they're just a response to taking a pill. And then a phase 4 study is what's called an aftermarket study. So that's a question that that's some other question, like does this medicine that helps the movement problems of Parkinson's also help the thinking problems of Parkinson's? That's called a phase 4 study. This is what happens, the Food and Drug Administration's new drug approval. The Food and Drug Administration is charged with answering the following question. Is the drug safe and effective? So the wording of the FDA is it requires substantial evidence of effectiveness demonstrated through controlled clinical trials. And the gold standard clinical trial is randomized. That means the assignment to one group or the other is random, like the flip of a coin. Double blind, which means the research participant and their family and the doctor who's running the trial doesn't know which people get assigned to the active intervention and which people get assigned to the placebo. Placebo-controlled means there's a placebo, are typically required. So sometimes if there's a very effective treatment on the market, the FDA will sometimes allow you to do a comparison between those two. But generally speaking, the drug companies don't want to do that because they have a better chance of showing their drug is effective compared to nothing at all, rather than something that's already known to be effective. And then the other thing the FDA is responsible for is deciding if the benefits outweigh the risks. All drugs have risks. And then they make sure that the package insert and the name of the drug, you can't have a name of a drug that sounds too miraculous, for example, because it's considered to be misleading. So they're involved with the package insert and the name and how the drug is dosed, et cetera. And then they're also responsible for determining if the factory is making the drug in a way that it's considered high quality, and then the whole transport chain. So if a drug needs to be refrigerated, for example, the drug company has to show how that drug will be maintained under refrigeration from when it leaves the factory to when it gets into your home refrigerator. So the FDA is involved in a lot of other kinds of stuff apart from just does the drug work. And the FDA spends a lot of its time policing those things with drug companies too. So the first step of a clinical trial protocol, so once you've identified something in animals, you've done the kind of preclinical testing, you have to write a protocol. So every research study has to have a protocol. And that protocol has several essential ingredients. So one is, what's the purpose of the study? I am going to study drug X to show if it improves walking and balance in Parkinson's disease. That's my question. Or I am going to try drug X to see if it improves thinking and memory in Parkinson's disease. And then there has to be a rationale. It's not something I just pulled out of thin air. The rationale is we've studied it in animals, or we've studied it in this, or some other kind of basic reason. And then the methods have to all be outlined. So the methods include the study design. So as I said before, randomized, like the flip of a coin, double blind, nobody knows who's on which treatment, placebo-controlled clinical trial. And then the study population. So who's going to be included? Are we looking at early disease, or are we looking at late disease? So for example, if I want to study something that improves thinking and memory, my patients who participate in the research better have problems with their thinking and memory. Otherwise, I'm wasting everybody's time. And then the other thing about inclusion criteria is in order to do research the most effective and cost-effective way, we need to select a group of people who are similar to one another. So a lot of times there'll be these criteria that people think are unfair, like your age or what other kinds of illnesses you have or what medicines you're already taking. But that's to try to make the group as similar as possible because it's easier to show change that's real change if the group starts out kind of similar and then you separate them into the two different interventions, the placebo and the active, and then that separation is easier to detect if they all started out kind of similar. Does that make sense? Do you have any questions about that? So it's not meant to be kind of Scroogey that we're not going to let you be in it. It's because we really need to be as effective as possible in the science. And then there are exclusion criteria. So if the medicine interacts with another medicine or if you've got a disease that's going to make it difficult to see if our treatment works, then that will be excluded. And then what the intervention is, what the dose is, whether it goes up, escalating kind of dose, what's the interval between doses, is it once a day, is it 3 times a day, is it a patch, is it an injection, does it go under your tongue, and then what the assessments are. And you have to have what's called a primary outcome measure, which means the one thing that you need to demonstrate, and that's because in the past people used to just measure 100 things. And then if one thing turned out positive, then they'd write their paper. But that's not really the way science is. In science, you say, I want to show that this improves walking. Here's my measure of walking. That's my primary outcome variable. I have a drug, I want to see if it improves thinking and memory. This is how I'm going to test the memory. That's my primary outcome variable. And then you can have secondary outcome variables too, but you have to have one thing in mind that's going to get better. And then you also have to specify how you're going to do the analysis. I'm sure you know the quote that there are lies, damn lies, and statistics, the 3 kinds of falsehoods. It's true that you can kind of twist anything around if you haven't specified ahead of time what that method of analysis is. This is the Institutional Review Board. This is a group at every hospital that does research has a group like this. There are diverse members. So there are people who are in the kind of area of study, like there'll be neurologists on the review board, but there'll also be cardiologists, and there might be basic scientists. There's always a community member. Sometimes it's a pastor, sometimes it's an advocate, sometimes it's a patient. And they read these protocols, and then they kind of talk about whether it's it's really to make sure that the people who participate in the research are safe, and also that it's worth doing. So if it's a kind of a dumb question and you're putting people at risk, the IRB will say no. And so they're involved in the initial approval, and then every year they have to review the study again, as long as the study's going on. If there have been any side effects that are unexpected, they review all those too, and they have the power to shut down a study, as does the FDA, if the investigators aren't doing a good job. So if you ever get approached about a study, there's this thing called the schedule of activities, and it's got all the visits or telephone calls, whatever the kind of contact is, and then everything that happens. An EKG, so an EKG may be done at baseline, and then at 6 weeks, and 12 weeks, and 24 weeks. Blood tests when they're done. Urine tests. Pregnancy tests for women who could become pregnant. They're all outlined on here, so that the research subject can see exactly what's going to happen at each visit. Yes? So usually it's different at every institution. I haven't been on the IRB at UW, so I'm not exactly sure how they get their community members. But I could have, do you come regularly to this or no? Yeah, I could see if Nancy could let you know what the particulars are here. But I was on the IRB for 5 years in Chicago, and we had a minister, we had an HIV community activist, we had a lawyer, we had all different kinds of people. The Parkinson's Disease Foundation actually has a patient advocate program where that's kind of a training thing, and there are several different aspects of that. One is kind of a buddy system for people, kind of a contact person if you want to be in research and have a little kind of support buddy. And I think they also train people to be on IRBs. It's a ton of work. It's a ton of work. I mean, these protocols are really big, and they're very boring. But it's a very good activity. So if you're approached for study, the next big thing is informed consent. And this informed consent kind of really rose out of the Tuskegee scandal where black men living in the South who had syphilis were not treated even after treatment became available, and they didn't consent. It was a terrible scandal in this country. And also the kind of atrocities committed by the Nazis in the interest of research, et cetera. So this kind of all rose as a way to protect subjects. And so there are certain critical elements of informed consent. Informed consent is both a document and a process. So if, for example, you were to call and say, I'd like to be in your study, we'd say, well, let's send you a consent form, and you see, and it has everything you need to know. So it says that it's research, it's not care. It says the purpose of the research, how long it will take, everything that's going to happen, all of the experimental procedures, whatever they are, any risks, any discomforts. So, you know, bruising in your arm from getting a blood test, for example. Fatigue if you have to take a lot of mental tests. All those things will be listed. Any potential benefits. And I have to say that participating in research, the purpose of that is to find new treatments. It's not for the people to get better. It's not a treatment, and it's really important to understand that. And sometimes if the intervention works really well, you do get better, but that's not the reason to participate. Any alternatives? So if I don't go to this research, is there something else I could take that might do, you know, might make me happy? Confidentiality is huge. So how will your confidentiality be protected? You don't want your insurance companies learning about research and stuff. How the records will be kept, and that the FDA can come and inspect those records, and they can look, and the FDA can look at your personal medical records too. Whether there's any kind of compensation. Do you pay parking? Or will you pay my transportation? Will you pay for my time? That's also involved there. Where to get answers if you have questions, who to call, what your rights are. So you have the right to withdraw, for example, from the study just because you want to withdraw from the study. What happens if you're injured, if you have a side effect from the medicine? The fact that it's voluntary and the fact that it won't affect your care at the center if you don't participate. So you have the right to refuse participation without losing any benefits of being a patient at the center. That you can, that's twice. You can refuse or you can stop, that's what it is. Who to contact, any anticipated risks to unborn fetus, so for pregnant women. Any reason that we could stop the study. And then any added costs that you might have by participating, any consequences. Let's say you decide you want to quit, but you need to taper the medicine. The consent form will say what will happen if you don't taper the medicine. A statement that if we learn something new that will affect your willingness to participate, so let's say you're in my drug study and then suddenly the cure comes out, I can't keep that a secret from you just so that you'll participate in my drug study. That would be considered bad. And then how many people will participate. In most studies, especially the phase 3 studies, are done at multiple centers, so like 40 different places in the country, each of which will recruit 20 patients. So this consent form will say, well, there'll be 20 people at UW, but there'll be 800 people in the whole country. Any questions about that? And when I say it's a process, you read the thing and then you have a discussion with the investigator, and then if you come in 2 weeks from now for your next visit and you have another question, then that discussion continues. So it's a process. Yep.
What is the purpose of the FDA being able to look at the individual participant's medical records?
Because one thing the FDA, what's the reason for looking at the individual medical records? Because the FDA needs to make sure there's no fraud going on. So there were people I even know, who would say that they had a study subject and they'd fill out all the data, but they didn't. Because you get paid per subject for the cost of running the study. And many people have been permanently banned from doing clinical trials because they did just that. So they have to have the person that goes with in the study records, no one's ever identified by name, it's by a study number. But the site will keep a list of the number and the patient, and then the study gets monitored, usually sometimes 4 times a year, sometimes even more. Someone will come out and make sure the data matches the stuff in the healthcare record. And then the FDA, and that's a person who's sent out by the study sponsor, it's called a monitor. But the FDA audits certain studies if they're worried about fraud, and also before approval, they'll usually audit to make sure that the data are accurate. That's a good question. Any other questions about that? So how can you learn about clinical trials? I love this website. So there's several different websites you can go to. This is clinicaltrials.gov. So in response to things like people not having a primary outcome measure and doing a bunch of different statistical analyses just to make things turn out right, all of the journal editors, the people who publish medical research, got together and agreed that they would require some kind of registration for all the clinical trials. And so our government set up this thing called clinicaltrials.gov. That's the website, clinicaltrials.gov. And you can go to that website, and every single clinical trial for Parkinson's disease in the world is registered on that website, because they can't publish their article unless it's been registered. And so this assures that they've identified what their primary outcome measure is, what they're going to look at, and then they have to say in advance what their statistical methods are. And so then what happens is if they write up their paper and then they send it to the journal and it doesn't match up with that clinical trials listing, then it won't be published. So that's a great thing for medical research, but it's actually a great thing for you because you can go on there and see what's happening. People ask me all the time, well, what's happening in Europe or what's happening here? So if you go to that website, you can type in Parkinson's disease in quotes, and it will show you on a map. So this was on December 27th. This is a listing, a visual listing of all the clinical trials that were recruiting subjects for Parkinson's disease on that date. And I'll show you what that interface looks like. This is what it looks like. This is the website, and right here, you can put in your condition, and you can put in other stuff. And then what happens is you get a list. You can put in, for example, only studies that have results already, and then there's a link to the results, for example. Or you can put in Parkinson's disease and constipation, or Parkinson's disease and dementia, and it will give you those trials. And then you can put in the ones that are recruiting patients. And then as you open each record, it tells you where it's being done. So a lot of them would be done in, Poland and England and 10 different places in the United States. And then if there's one close to you, you can kind of click on that place, and it will tell you who the contact information is. So in fact, if something looks really interesting, many studies, especially for rare conditions, they'll pay for you to go. So for example, I had a patient with a different condition who was going to Toledo, Ohio, from Rockford, Illinois to Toledo, Ohio, every 6 weeks for a drug study. And he and his sister, who traveled with him, had their airfare paid and all their transportation and their meals and their hotel to participate in that. So then there's that contact information you can call. It's very nice. But mostly it gives you an idea of what's happening where. And so this is from when I looked on December 27th, and so these are some of the ones that were listed on there. So an injectable medicine called liguritide, and a randomized double-blind placebo-controlled study of liguritide, phase 2. So remember, that's the kind of more early phase study. Varenicline, this is one that's been done at Rush, looking at falls in Parkinson's disease. Here's another one for gait, balance, and falls. Cannabidiol. A lot of people are interested in marijuana and its substrates or its metabolites for Parkinson's. So that's an actual study of cannabidiol, which is a marijuana derivative. And then an implant of verpinerol. That's another one. And so I have here what the places where it's being done too. And so those are symptomatic treatments. And then here, disease-modifying or neuroprotective treatment. So this is an experimental. A lot of times experimental drugs won't have a name. They'll have a number, BII-B054, intravenous in Orlando and Tampa, Florida. Here's another experimental one in Germany, London, and a couple of United States places. Nilotinib, I'm going to talk a little bit in a minute about, which is a phase 2 study that's funded by the Michael J. Fox Foundation. So it just gives you kind of the ability to see what's going on and if anything interests you and if you might be able to go there. Let's say you've got a daughter that lives in Phoenix and you want to go there. That would be a nice thing. Now, tell me if you've ever heard something on the radio or on television that said, like, this could be a cure for Parkinson's, right? Have you ever heard any of those? Because it happens. And you need to be able to be critical in evaluating the accuracy of those reports. So here's a little bit of few little tips about that. So, oh, come on, baby. See, if I just get close to it, it knows. So several times I've been asked about exenatide. Has anyone heard about exenatide? So here's the media pitch on exenatide. The trial on 62 patients published in The Lancet hints, that doesn't sound like a very scientific term, that medicine halted the progression of the disease. Now, does that not sound like a miraculous drug? Does that sound wonderful? So here's the actual study. So this is a phase 2 study, and you know this because the number's only 62, and a phase 3 study, one that would be required for FDA approval, is going to have hundreds of patients. They had moderate Parkinson's disease. It was a randomized, double-blind, placebo-controlled study. That's the best kind, right? And it was an injection of this exenatide, which is a drug that's already on the market. And it was a 48-week study with a 12-week washout. And here's the data that was presented in the paper, okay? So the primary outcome measure was something called the UPDRS, the MDS-UPDRS, which is a rating scale that assigns scores of severity to a bunch of different tremor measures and rigidity and slowness, and then you get a score. And these are the data. So the blue here, and so these are the weeks, 0 weeks to 60 weeks, and the blue here is the exenatide. So they started out better. So in this thing, actually, a higher score is worse. They started out worse than the placebo group. But at the end, they were different. So the placebo group was actually 2 points worse, and the drug treatment group was 1 point better than they started out at the baseline. So because down is better and up is worse. So that looks pretty, that looks like they're separated, right? Right? So one thing is, how are the data presented? So, and that's, in that graph, they didn't extend the scale all the way down to 0. So they had, so what happens if you extend the scale all the way to 0 is those two curves don't look very different after all, right? And then if you include the full range of the scale, so this is a scale that goes up to 135. If you include the full range of the scale, now they don't look different at all. Right? So partly, if the way you graph your results can make something look a whole lot better than it is. So that's misleading. And then the second thing is, even if a difference is statistically significant, it may not be clinically significant. So this MDS-UPDRS score has been analyzed, and it's an improvement of 3.25 points is considered clinically significant. In other words, the patient can tell they're better. That's what clinically significant means. Not clinically significant means that I can tell they're better, but they can't. So their score is 1 point better, or score is 2 point better, but the patient doesn't feel it. So you need to actually have 3.25 points improvement in order for the patient to feel different. You saw there was only a 1 point improvement on that scale. And you have to have 4 points, almost 5 points decline before the patient knows they're worse. So those results, which look great on the first slide, not so great on the second slide, now seem to really not be very relevant at all. And then statistical significance is really a number that expresses how sure you are that the curves are different. That's all. How sure you are that the curves are different. So it's the probability that the result could have occurred purely by chance. And if it occurred by chance, then you're not sure of the difference. Does that make sense? Got that? Any questions about that? So usually we consider something statistically significant if there's a less than 5% chance that the results occurred purely by chance. So you can have statistical significance like in this study, but not have any clinical significance. Any questions about that? Okay. So that's one way a study can be misleading. So here's another way. So the media's interpretation of this result was the trial of 62 patients, you've already heard this, published in The Lancet, hints the medicine halted the progression of the disease. If you go back to the paper and see what the authors said, they said, compared with placebo, exenatide treatment is associated with positive and persistent effects on the score. They didn't say clinically significant. Whether this drug acts as a novel symptomatic agent, so just like a dopamine drug makes the symptoms better, influences compensatory responses or behaviors, or has neuroprotective effects on underlying pathology is unclear. So the media took that statement and turned it into hints that medicine halted the progression of the disease. So partly it's that the media is only interested in your story if it's a miracle, not if it's a statistically significant but not clinically significant difference. So part of it is the media's fault, really. And then I want to talk a little bit about another kind of misleading study that's by the design. So you saw, I'm going to show you a very small study, but even that study was pretty small. So when you design a study, you don't want to waste all your money by not having enough people in your study. And so the statisticians actually have an extremely complicated formula. You go to them and say, here's my primary outcome measure. It's the MDS-UPDRS. How many people do I need to have in my study using that measure in order to show statistical significance? And so they plug the numbers in so they know, for example, what the variation is in that scale by published studies. So we know if you have 100 people, they're going to have a certain amount of variability in that scale. And then we know we want to have a power, so that's a number that says, if I have this many people in my study, I have an 80% chance of demonstrating a difference, a significant difference. So you plug that number in. That's basically what you want it to be. The effect size. So you say, well, I think this treatment's going to make them 20% better. So that's the effect size. And then you plug in that level of statistical significance into this formula. And that formula generates what's called a sample size. And as I told you, in a large study of neuroprotection, it's going to be in the hundreds, usually 500 people. It's a good ballpark for a Parkinson's disease neuroprotection clinical trial. So here's a study that got a lot of press. So who's heard of nilotinib? This came out in 2000. Yeah, okay. So nilotinib is a marketed drug. It's used to treat blood cancer. It's the chemotherapy agent. In preclinical studies, it actually helps the cells, the brain cells, clear protein out. Kind of sounds like just what the doctor ordered, right? So this group at Georgetown, actually a good group who'd done some of the preclinical work, decided to do a study. So they took 12 patients. Does that sound like 500? 12 patients. They did an open label trial. That means no placebo, no randomized, no control at all. They put all 12 patients. And it was a mix of patients. So some of them had Parkinson's disease with dementia. Some of them had diffuse Lewy body disease, which is a related but not exactly the same condition. So now they've mixed patients. And then they broke them up into two groups and gave some, 5 of them 150 and 5 of them 300 milligrams. And they studied them for 24 weeks, and then they stopped them, and then they saw them at 36 weeks. So they said that there were beneficial effects, and the beneficial effects reversed when they stopped the medicine. Does that sound like a neuroprotection, or does that sound like a symptomatic treatment? When you go right back to the way you were before after you stop it. They used both movement and cognition measures. They did say that it should be interpreted with caution and that further testing was needed. So these are the things. So it's only 12 people. It was open label study. It reversed when the drug stopped, so it wasn't really a study of neuroprotection. And here's the media. Researchers report first therapy appearing to reverse decline in Parkinson's disease. Does that sound like what we just talked about? No. Is that the whole value? Well, I think what happened here was an overabundance of enthusiasm and without stopping to think. And this kind of a study where there's a marketed drug and it's the investigators kind of their wild idea, the FDA doesn't have a lot of, they would never allow a drug company to do a study like this. In a million years, the FDA would not allow this. The question is why their IRB allowed it, and I can't answer that. But yes, if you believe in something, it's like the magic feather in Dumbo, right? If you believe in it, you can design your study to make it look good.
But I think in some situations you use hints and you use the word appears. Anytime you use words like that, it's not the same. They don't know.
Right, but then if you go to the Georgetown website and watch the interview with these doctors, They pretty much pitched it to the media as if it was, yeah, as if it was researchers report first therapy appearing to reverse decline. Oh my goodness, did they believe this? I still get questions about it. So, Georgetown. It's a good place. It's a good place. But so the Michael J. Fox Foundation has ponied up the money to actually do a phase 2 study, which has 135 subjects in it, and they will have the answer. The reason they're doing the phase 2 study is they don't believe this initial hype, and they want to just put it to rest because people are interested. Obviously, a drug that's on the market that reverses the decline and progression, who wouldn't want that? So there will be a study that's done that will have information about statistical significance and clinical significance and toxicity, et cetera. So, yeah. Yes? This got published in a good journal, not a great journal, and it was widely derided by the research community when it came out. You can look at it, and there are, like the Parkinson Disease Foundation and the Michael J. Fox Foundation, their websites will address these kinds of media releases so that you can get the more balanced view. But having been someone who's kind of been involved with media reports and research, I know that they really want you to bring in a patient that's benefited from the research. And remember, I told you we don't do research for individual patient benefit. We do research to find things that work. They'll want to bring in a miraculous patient. They'll want to make you say words that are more than the study does. So it can be very, they're not interested in things that aren't, that they can't put on that, trailer before the news starts. So you have to be careful. But there are sources to help you. And you just need to have just a normal. And here are the data, right? I actually went to the paper and I looked at all the scores, and here are the data. Does this look like something that slows decline? So each one of these is an individual patient. Oops. Each one of these is an individual patient, and it's every 4 weeks, and at the end they stop the medicine. And when you stop a medicine, just like the placebo effect, when you take a medicine you feel better, when you stop a medicine you feel worse, so generally speaking. But there's really not very compelling, I think. There's this one person who dropped from a score of 42 to 4, which I think must be an error, because that can't happen, and then went back up again to 39. So if you, and they didn't show this data in the paper. I plotted this data just using what was published in the paper. So are you convinced? No. And then finally, I get asked all the time about stem cell. My daughter really wants me to go to the Stem Cell Institute, and it's $13,000, and they're going to take stem cells from my fat, they're going to inject it into my bloodstream. I've gone to the website, I've seen the videotapes. And so I've taken this term baloney detection from Carl Sagan, who had a whole thing on detecting baloney in scientific research, very good, online. And this is the way to know. So on these websites, the treatment is good for what ails you. So if you've got arthritis, fibromyalgia, headaches, Parkinson's disease, Alzheimer's disease, everything is fixed. It's a cure-all. So that's one sign that it's BS. The second one is all the evidence is personal testimonial. So click on this videotape of Reggie before and Reggie after the stem cell treatment. But there's no data, just testimonials. They're quick fixes, so it's easy. They're going to take my fat out and turn it into stem cells and just inject it into my vein. It's a quick fix. A lot of times all natural attracts people. So it's kind of a baloney detection. Anything that's miraculous or uses the word cure, that's baloney. If they're conspiracy theories, like this is a secret that we know, but your doctor won't tell you about it because they really don't want to cure Parkinson's disease because that's how they make their money, seeing people with Parkinson's disease. As if the person who cures Parkinson's disease isn't going to have a lot of opportunities in their future. And then finally, you have to pay for it. So if you participate in research, you never have to pay for it. Research is free. The sponsor's paying for the research. So if they're asking you, and they've recently come up with a little bit of a scam. So for example, when I was looking at the website for clinical trials, I saw one of these scam stem cell places was on that list. So I called, and it said they were doing a study. So I called them up. And I said, I'm interested in your study. Is the treatment free? And he said, oh, no, the treatment is $16,000. And I said, well, then how is it a study? He said, oh, we're collecting data on people who've had the $16,000 treatment. And that's not a real study. So, but they had that registered on clinicaltrials.gov. So now you have to even be a little bit more careful about that. But you can always call, and they'll tell you if it's $16,000. I can think of a lot of things that you could better spend your money on, $16,000. So I just wanted to say the 2 studies that we're going to be starting here, neither one is approved yet, so we're not recruiting. One I'm really excited about. This is Dr. Bullion Dent is the investigator for this study, and it's the subcutaneous levodopa pump. So, you know, one of the problems with as Parkinson's disease goes on is the medicine schedules have to become more complicated because the disease is more severe, and you have this thing called on-off or dyskinesias. And so stabilizing the amount of drug in the system helps that. And so this would be a continuous pump. There already is a pump on the market where you've got a tube in your stomach and it pumps into your small intestine, actually. A lot of people don't want a tube hanging out of their stomach, and so that's not been a very popular therapy. But this would be sort of like an insulin pump where the needle goes into the tissue right under the skin. I think that's very exciting. And then another one, so remember we really focused on the movement aspects of Parkinson's, but not a lot of attention on the other parts. So this is a new agent to improve thinking and memory in people with Parkinson's. So those 2 will be starting, I think, within the next couple of months. And my goal, when I was in Chicago, we always had at least a dozen studies going. And there's a lot to benefit. You know, when you're in a study, you spend a lot of time with the researchers, which is good because you learn a lot about the disease. It's just it's a very nice kind of interaction with patients and family members. And you also get that kind of empowered feeling that, you know, let's face it, none of you deserved Parkinson's disease. None of you asked for it, and none of you did anything to bring it on. But it's a way to kind of fight back against this kind of random thing that struck you down in your golden years. So there's a lot of positive benefit for participating in research studies. And the Parkinson's Disease Foundation's done a little study to show that research participants do better. They have better outcomes than people who don't participate in research. So there are lots of reasons to participate in research.